All right, biologists, let's take a look at proteins, subtopic 2.4 of the topic molecular biology of the IBDP Biology standard level. Now, proteins are essential in any organism. They perform a wide range of roles. There are structural proteins that give your cells their shape. There are proteins in your extracellular matrix. There are proteins that accelerate chemical reactions. There are proteins that are messengers. There are proteins that are hormones. There are proteins everywhere. And proteins are made of amino acids. So this is one of the things you have to know how to draw in the test. This is a generic amino acid. And this carbon here, so we can start with this carbon, and it's linked to 4 different things. This carbon has a special name. This is the alpha carbon. Okay, so the alpha carbon is connected to 4 different things. The first one we're going to see is this green group here. So you see it has nitrogen and 2 hydrogens. This is the amino part of amino acid. So it is connected to an amine group, nitrogen and hydrogens. It is connected to this yellow group here, COOOH. This is a carboxyl. This is a carboxylic acid. So this is the acid part of amino acid. So carboxyl COOOH linked to the alpha carbon linked to an amine. We also have a hydrogen here every time, and this other R group. The R group or radical R group, it is variable. So this is what makes each type of amino acid different from other types of amino acid. So every single amino acid has the amine group, an alpha carbon, and a carboxyl group, right? And a hydrogen here that doesn't do much, just stays there. And then we have the R group, and each different amino acid has a different R group, which gives that amino acid their unique properties. So let's take a look at this amino acid here. How do I know this is an amino acid? Well, here we have, let's use the same colors. We have here the amine, right? So here we have in this ball and stick model, nitrogen and hydrogens. Nitrogen usually blue, hydrogen usually white. We have the COOOH. We have our carboxyl here from the carboxylic acid, the acid part of amino acids. So as usual, oxygen in red, hydrogen in white. We have, of course, the alpha carbon connecting both. So here we have the alpha carbon. We have 1 hydrogen right here. And the R group in this case is another hydrogen. The R group in this case is another hydrogen. Oh, how do I know if it's this one or that one? Doesn't matter. In this case, both are hydrogen. All we know is the R group, the radical, the R variable group. In this case, it's a hydrogen that makes it a glycine. So glycine is this particular amino acid here. Let's see another example. This guy, slightly different, but still we have our alpha carbon here. Now remember that carbons are the vertices and the tips. So we have an alpha carbon here, which is this carbon here, alpha carbon, connected to an amine group, carboxyl here. Where is the hydrogen? Well, in this form, this representation, the style of representation where the carbons are only the vertices or the tips, the hydrogens are not even mentioned, except, of course, if they are connected here to the nitrogen or the oxygen. So the hydrogens here are implicit. You don't have to draw it, but it is hiding right here behind the carbon. And the R group here is a methyl. It's a methyl CH3 group. So this is a CH3 This amino acid is alanine. Oh, do I have to know the names of the amino acids and what they look like? No, you don't. There are too many, and there's no point in memorizing what they look like. But you do have to be able to recognize one and draw a diagram of a generic amino acid. So here, again, same thing, alpha carbon, carboxyl, amine, hydrogen hiding here, and this very large R group. So the size of the R groups will interfere in the protein. The fact that this is made of carbons and hydrogens will interfere, or it could be made of something else. Here in this case, it has a carbon, but also has sulfur. It's a different amino acid. Sulfur is polar, and that will make all the difference. All right, so amino acids are linked together by condensation to form polypeptides. Polypeptides are chains of amino acids, right? It could be used as a synonym for a protein if you're dealing with only one chain. So let's draw here what these molecules are representing. All right, so here I have these two amino acids. I have this guy here, amino, alpha carbon with two hydrogens, so alpha carbon with two hydrogens, and carboxylic acid. And the same thing here, amino, alpha carbon with two hydrogens, and carboxylic acid. So what happens here, the reaction is called condensation. Look at this OH. They come close together here with this NH2, and the oxygen looks, well, I have a hydrogen, and the other guy over there has another hydrogen. But if I had two hydrogens, I could make a water molecule. And this is exactly what happens. See this water molecule here? It comes from this oxygen looking at this hydrogen here and thinking, well, if I had another hydrogen, I could make a water molecule. And they just pop off. Just see here, you have C double O, and then straight nitrogen with one hydrogen here. It's this nitrogen with this one hydrogen that is left. So when you have two amino acids and they are going to be linked together, this is what we call peptide bond. Here we have a peptide bond between the carboxyl from one amino acid to the amine of another amino acid, and you have this water molecule that comes out of it. So this here would actually look more like this. We would have our peptide bond here. Look at that. Good old peptide bond between carbon and oxygen here, C double O, and nitrogen, hydrogen here. It's this guy, okay? It's this guy. And what about the other OH and the other H? They make a water molecule. Oh my God, this is super complex. Yes, but also the same thing. Look here, you have C double O OH, NH2. Well, this is carboxyl, this is an amine, and you know what that means. It means water time. It's water time. See, you have a OHH, oxygen, hydrogen, hydrogen, oxygen, hydrogen, hydrogen. They are dying to make a water molecule, and they do. Here we have nitrogen straight to carbon double O, nitrogen straight to carbon double O, and this is our peptide bond. So every time you have the condensation between two amino acids, you have one water molecule. Do we have a general rule for that? Yes, we do. the number of water molecules that are made by condensation is the number of amino acids minus 1 every time, which is the same as how many water molecules do you need to hydrolyze. So the opposite of condensation is hydrolysis. How many water molecules do you need to hydrolyze a peptide? Well, it's the number of amino acids in that peptide minus 1. So if you have 20 amino acids, you need 19 water molecules. Okay, and how many amino acids do we have to make our proteins? Well, there are 20 different amino acids that you can combine in all sorts of ways. Of course, proteins do not only have 20 amino acids, they have way more than that, but you have 20 different amino acids. It's just like the alphabet of proteins. Now, the words you can write with that alphabet, they vary in length, they vary in the combination of letters you use, but the alphabet here has 20 letters, 20 different amino acids. Out of these 20 amino acids, 9 are hydrophobic. So they do not like water. Why do you have hydrophobic amino acids? Because hydrophobic amino acids, they try to stay away from water, and then the protein shape will be determined, for example, by these guys trying to avoid water and folding inside the protein. Or if you have a protein that needs to cross the membrane, a membrane protein or a membrane channel, or it's a protein that needs to be anchored in the membrane. The membrane has a hydrophobic part, so you need hydrophobic amino acids to cross the membrane or to be anchored in the membrane. 11 amino acids are hydrophilic. That means they like water. Out of these 9, 3 are aromatic. 3 are aromatic. That means they have rings. They have carbon rings in them. And 6 are aliphatic. That means they do not have rings. What about the hydrophilic? Well, the hydrophilic, 4 of them are, well, all of them are polar, but 4 of them are not charged. They do not have electric charge. They do not have electric charge. They are polar, but they do not have electric charge. And 7 of them have electric charge. They are ionic. Out of these 7, 4 have negative charge and 3 have positive charge. You have to know exactly how many of each type. No, you do not. But you've got to understand that we have all these flavors. We have some of them hydrophobic, some of them hydrophilic. Some of them are aromatic, larger probably than the others. And 6 of them are aliphatic. They're non-aromatic. They do not have the rings. Some of them are polar, but they're not charged. Those are going to be the guys with sulfur, with oxygen. They are going to give this hydrophilic feel to the amino acid. And the others are ionic. You're going to identify them by the charge they have, negative charge or positive charge. And again, all of this will interfere with the shape of the molecule. The polar amino acids, they like to be in contact with water. The negative amino acids want to be near the positive amino acids. So the molecule, the protein will fold so that the negative will be near the positive. At the same time, the positive wants to stay away from the positive, negative will stay away from the negative, so on, so on. So here's an overview of the amino acids. You see that here we have the aliphatic. So here we have just CH3. Here we have carbon and hydrogen only, but not no rings and nothing polar. Here we have the rings on the aromatic ones, and all the rest will be polar. They have here charges. They have nitrogen, they have oxygen, nitrogen. Some of them might contain sulfur. Pay attention to this guy here, cysteine. Cysteine is an important amino acid. We're going to see why later on. Do you have to memorize all that? Of course not. Right, so from these 20 amino acids, some of them might be modified. So you can spice up your protein by modifying some amino acids, which is the case of a very important protein called collagen. So collagen is a protein that is only found in animals. It is a very tough structural protein that you find in connective tissues. A lot of people think of skin when they're talking about collagen, and it is true, you have collagen in the connective tissue under your epidermis, but you also have collagen in your bones, you have collagen in cartilage, you have collagen in ligaments. It's a very, very important and very, very common protein in animals. Collagen is what is used, for example, to make gelatin and gummy bears, unless they're vegan. And one of the things that make collagen so tough is this amino acid called hydroxyproline. The thing is, it is not one of the 20 amino acids we saw in that list. So how do we get it? Well, we originally start with proline, this guy here. And then we have an enzyme called proline hydroxylase. And what does that enzyme do? Well, it hydroxylases proline. It will add a hydroxy to the proline. So it's a very descriptive name. Proline hydroxylase adds the hydroxyl here to the proline and makes hydroxyproline. And that tiny difference makes all the difference in the world. Think of pirates, for example. Think of pirates and their smiles. You're gonna think of a pirate and you always think, well, besides the eye patch and the parrot, you think of missing teeth. Why is that? Well, think of the life they had, long distance navigation or long time in the sea. What do you have in your ship? Well, you have rum and wine and whatnot. You have a little water. You have some curated meat and whatever you can catch. You know what you don't have? Fruits and vegetables. One day, a doctor in the British Navy decided to put citric fruits in one of their ships. He noticed that the crew in that ship would not suffer from teeth loss like other crews would. In a way, that was a controlled experiment that showed that citric fruits in the sailor diet would prevent them from losing teeth. And from that point onward, citric fruits were a staple in the British Navy. Now we know why that is the case, because citric fruits have vitamin C, which is an important cofactor for proline hydroxylase. So without vitamin C, you cannot have hydroxyproline, and then you cannot have collagen, which, as we said, is an essential protein in connective tissue, including the tissue that connects teeth to bone. So to keep our teeth in place, we need vitamin C to make collagen, to make the tissue that holds our teeth in place. Now, every organism, archaea, bacteria, or eukaryotes like ourselves, all of us have the exact 20 amino acids to make all our proteins. Think of all the thousands of proteins that an organism might have. Think of the millions of organisms that exist. Think of the billions of organisms that have ever existed since the beginning of life. All of them, as far as we know, they use this 20 amino acids, the same set of 20 amino acids. This is because the set contains enough diversity so you can have all sorts of proteins, but also it's an indication of the origin of modern organisms, not necessarily the origin of all organisms that have ever existed ever, but at least it's an evidence that points to the existence of LUCA, which stands for last unicellular ancestor. It would be the organism that gave origin to every other organism still in existence, had these 20 different amino acids, and this is a shared trait between this common ancestor and all of its descendants. And we can combine all these 20 different amino acids in any possible ways. Like imagine if you have only one amino acid in a sequence. Well, it's not really a sequence, just one amino acid. So there are 20 of them. So it's 20 to the power of 1 combinations, which is exactly 20. If you have 2 amino acids, well, imagine here, 2 amino acids. That means I have 20 possible amino acids for this first position times 20 different amino acids for the second position, which is 400 or 20 to the power 2. What if I had 3 amino acids in my sequence? Well, that's 20, 3 times, that's 20 to the power 3, which is 8,000. If I have 4 amino acids, well, then you guessed it, it's 20 to the power of 4. Not 204, it's 20 to the power 4. And that is 160,000 possible combinations. 20 to the power of 6, that's 6 amino acids. And that alone is 64 million combinations. Now, how many amino acids do you need to make more than 10 trillion combinations? Believe it or not, 10.24 trillion is 20 to the power 10. With only 10 amino acids, you can have more than 10 trillion different proteins. Now, remember that proteins don't only have 10 amino acids, they have hundreds of them. This is why we can have such diversity that we can't even comprehend. It's too large a number for humans to understand fully what that represents. You can write this number down, but you can't really understand what it means. It's insane. This is why 20 different amino acids is more than enough to give origin of all diversity we have now and in the past. What about this amino acid sequence? Let's talk more about it. So this sequence will determine the 3D conformation of a protein. We have basically 3, sometimes 4 different structures of a protein, 3 or 4 different levels of structure in a given protein. The first level, the primary structure, is just the sequence of amino acids. So if you change the sequence, you change the primary structure. The primary structure is determined by the sequence of amino acids, and the amino acids are linked by covalent bonds, which we call peptide bonds. Now, if you remember, the peptide bond is the bond between the amino group of one amino acid and the carboxyl group of another amino acid. The secondary structure, we have 2 conformations here. Sometimes this chain of amino acids will spontaneously turn into a helix. We call this the alpha helix. The alpha helix. Or it could form zigzags, and we call it beta sheets or pleated sheet. This structure is maintained by hydrogen bonds. So here we have hydrogen bonds. Hydrogen bonds are weaker individually, but we have so many of them that it makes the structure very stable. These hydrogen bonds happen between the amino and the carboxyl groups of different amino acids. So you can have hydrogen bonds happening here between the amino and the carboxyl group of different amino acids. Same here, the amino acids parallel to each other here. We have amino on one side, carboxyl on the other side, and hydrogen bonds between them, stabilizing these structures. Primary structure, no R group is involved here. Secondary structure, no R groups are involved here. Now the tertiary structure is the 3D conformation. Think of tertiary, think of three. Three what? 3 dimensions, okay? It's when you have your alpha helices and your beta sheets, and they fold over each other, forming this 3D structure that we have. So you can have the helices here, and you have another helix here, and you have another one, and you might have, I don't know, a beta sheet here and there, forming the 3D structure. And this is where the R groups are super important. You have R groups determining the 3D structure, the R groups determining the tertiary structure of a protein. And why is that? Well, because this part here is kind of hidden in the center of the protein. So probably you're going to have hydrophobic amino acids here. These ones out there, you have the hydrophilic amino acids. You have a large area here, easy to accommodate large R groups, but this tight loop here, you might need smaller R group amino acids. So there's different combinations, and this is why the primary structure is very important, because this is why the primary structure is going to say, hey, about here, I'm gonna have only small R group amino acids so I can make this fold. Or about here, I'm going to have hydrophobic amino acids so they can fold inward inside the structure hidden from the water. In the end, it all comes down to the sequence of amino acids, to the primary structure, because the amino acids in the primary structure will have here their R groups that will give the tertiary structure, the 3D conformation, the 3D shape of the protein. And this is where most proteins end. Now, some proteins, they have more than one peptide. They have more than one piece that combine and make a larger structure. It's a combination of several peptides coming together and making a larger structure, a larger protein, and that is the quaternary structure. The quaternary structure is made of more than one peptide working together. So here we have one peptide in purple. We have a different one here in yellow. We have another one here in magenta and another one here in cyan. We have 4. It doesn't have to be 4, okay? Quaternary means 4, but you don't have to have 4 peptides to be quaternary. You just have to be more than one combined to make this protein. And the protein is made of more than one peptide. In this case, we have a very famous protein called hemoglobin. And hemoglobin is the protein that carries oxygen. This is why your blood is red. It's the protein inside the red blood cells that carry the oxygen. You see this thing here? This is, in the case of protein, is called a heme group. Now, the heme is not made of amino acids. It's a different molecule, and inside the heme group, you have an atom of iron. This is why people say, oh, if you don't eat enough iron, you're gonna have anemia. What does that mean? Well, you do not have enough iron, then you do not have enough heme. Without heme, you can't make hemoglobin, and you can't carry oxygen, and that's not good. Since the heme is not made of amino acids. It is a part of a protein that is not made of amino acids. We'll call this a prosthetic group. Prosthetic comes from prothesis. It needs to be there, but it's not made of amino acid. So it's called a prosthetic group. Many proteins have different prosthetic group. Not all the proteins have one. Most proteins don't have one, but some of them do, and hemoglobin has a prosthetic group called heme. Heme is a prosthetic group which contains an atom of iron. And it's to this atom of iron that the oxygen molecule binds. But you still need the rest of the protein to make the whole thing work. All right, so let's do it again. This is an alpha helix. An alpha helix is a secondary structure. So here we have the amino acids. It forms this helix. This helix is supported. This helix is stabilized by the hydrogen bond. See here the hydrogen bond? What we have the oxygen and a hydrogen here making this hydrogen bond. Look at them. Oxygen, hydrogen, nitrogen. So you have here the oxygen, hydrogen, nitrogen. And this hydrogen bond here, which is an attraction between the partially positive hydrogen and the partially negative oxygen, this is what stabilizes the alpha helix. Same for the beta sheet. Here we have our beta sheet, which is also secondary structure. And here we have nitrogen, hydrogen, oxygen. So they are parallel, and hydrogen bonds, these hydrogen bonds, are going to stabilize the beta sheet. So secondary structure is stabilized by hydrogen bonds between the amino group and the carboxyl group of amino acids. Okay, in yellow here, you have the R groups. See how the R groups are not part of this structure. The R groups are doing nothing to stabilize the beta sheet. The R groups do not take part here. Primary structure is the sequence of amino acids, and then it's going to fold into helices and sheets, and then that is going to fold again into the tertiary structure. Here we can see how the R groups can interfere in the tertiary structure. One important thing that we have is the disulfide bond. It's this bond here between cysteine amino acids. I said it was important, and this is why. You have the cysteine here, another cysteine over there, and they can, because they have this SH group, the SH group of the cysteine, they can bind covalently. They can bind with a covalent bond, which is not breakable, and it's going to stay like this. It's going to stay like that. So it's a pretty permanent bond. Are the others not permanent? Not really. You can break a tertiary structure. You can break a tertiary structure if you add too much heat, or if the pH is too acidic or too alkaline. Okay, if the pH is too acidic, so here, too acidic, too alkaline, or if you heat up the protein too much, you are going to break the tertiary structure. You're not going to break the secondary structure, and you definitely are not going to break the primary structure. But the 3D shape of the protein will be ruined if you heat the protein too much. Think of egg whites. Egg whites, when they are raw, they are transparent and liquid. They are fluid. And then you fry an egg. You put too much heat in that egg white. What happens to the egg white? Now it is white. It's not transparent anymore. And it's solid. It's not a fluid anymore. So we are changing color and physical state just by denaturation. Denaturation is when you lose your tertiary structure. It is not reversible. Can you make the egg white raw again? You cannot. What if, but it's too much heat. What if I put the egg back in the fridge? Not going to work. Once you denature the protein, it doesn't go back. Okay. Once you denature the protein, it does not go back to its original tertiary structure. So the tertiary structure is determined by hydrogen bonds, covalent bonds, like the disulfide bond, ionic interactions between charged amino acids, dipole bonds, hydrophobic, hydrophilic interactions, all that between the R groups of the amino acids, between the R groups of the amino acids. And if you heat up the protein, or if you put the protein in an environment that is too alkaline or too acidic, you're going to denature the protein. You're going to have protein denaturation. That means you're going to break the tertiary structure and it never comes back. This is why fever could be dangerous. Fever is the body mechanism of rising the temperature so the pathogens, proteins cannot work anymore. The problem is if you have a very high fever for a long time, your proteins are going to start to change, to lose their tertiary structure, and then they're going to lose their function, and then it's not good for you. So fever is like your body playing chicken with a pathogen. All right, let's take a look at the two main categories of tertiary structure. Proteins can be either globular, like this, in which, you know, they are globular. They're not too long, they're not too short, basically the ball-ish shape of the protein. Or they could be very long, like a fiber, which is the case of fibrous proteins. Rule of thumb, these guys are soluble. These guys are not. So these guys are structural proteins, and these guys are not. Again, rule of thumb. Example of a soluble globular protein. Enzymes, DNA polymerase, helicase, RNA polymerase, trypsin, amylase, rubisco. All these enzymes, they are globular proteins. Fibrous proteins, our friend collagen is a fibrous protein. Collagen, actin, all of them are fibers. Fibrin, for example, they are all fibrous proteins, not soluble in water as you want structural proteins to be, because if they are soluble, well, they're going to dissolve in the cytoplasm, they're going to dissolve in the extracellular fluid, and they're not going to be good structural proteins. But globular, they are important to be soluble. It is important that they are soluble because they have to diffuse through your water in the cytoplasm or from one cell to another. So that's important that they are soluble. And what determines the sequence of the amino acids and the peptides? Well, it's all in your genes. In a nutshell, because we have a subtopic just for that, but in a nutshell, you have your DNA, and DNA is like a recipe book. So if you have a recipe book, the DNA recipe book, it has the recipe for making all your proteins. But for eukaryotes like you and I, the DNA is inside a nucleus. Okay, so it is locked inside a nucleus. It's too big to leave. And the guy who cooks the proteins, the guy who makes everything, he has a nice mustache, which is not so nice. The chef of the restaurant, the guy who prepares all the recipes, the ribosome, is in the cytoplasm. So you need a copy of the recipe, a copy of the gene to reach our friend ribosome. And this copy is the messenger RNA. As the copy of the message has the copy of the gene, the recipe, so that the ribosome can read that message, can read the recipe, and make a protein. How does it know which amino acid to add and which amino acid not to add? Well, it's all in the message, it's all in the DNA, it's all in the genes. That means if you change the gene, a mutation, for example, you might end up with a different protein. And as we said, we have tons of different proteins with all sorts of functions in all organisms. Some examples of proteins are enzymes. Enzymes catalyze reactions. They make reactions happen faster. And the most common enzyme in the world is an enzyme called rubisco. It is involved in photosynthesis. Some hormones are proteins. Now, each hormone has a different function. Not all hormones are proteins, but some of them are. For example, insulin. Immunoglobulins, which is the same as antibodies, they are globular proteins. So antibodies, also known as immunoglobulins, sometimes you're going to read something about, oh, this person had antibody test and tested positive for IgG or IgM. And you're like, wow, what does that mean? Well, Ig is immunoglobulin. You have 5 types of immunoglobulin, 5 types of antibodies, but we're going to talk about them later, not now. So antibodies involved in defense, in specific immune response, they are proteins as well. Some proteins are pigments, like the ones in our eyes that interact with light, rhodopsin, or hemoglobin, that gives our blood that nice red color, or structural proteins. We talked about collagen, but also spider silk or silkworm silk. They are all proteins. And every individual has a unique proteome. Just like we have genome, which is the set of genes of an organism, each individual, each cell even, has a different proteome. It's a different set of proteins that are expressed. Like we learned from the gene, we have the messenger RNA, which could be spliced in different ways. Okay, so you can have from one gene, you can have different messenger RNAs, and then these RNAs will be expressed into proteins. You can analyze a proteome using a technique called electrophoresis. So basically, you have this gel here in a solution. You apply an electric field to the solution, and in these wells, in these pits here, you're going to put your protein. So you're going to put the protein here, and the electric field will make the protein travel from the negative pole to the positive pole. But since this gel is kind of like a mesh, it will act as a filter. The smaller proteins will travel faster, and the bigger proteins will travel more slowly. So you can separate the proteins by their size. So we'll see the result. It's something like this. You have heavier proteins. So here we have this protein here, which is heavier than this one, which is the same as this one. They are lighter than that guy. And usually you can have here like a scale. You know exactly how much, each of those weight, the weight of the proteins measured in kilodalton. So you know the size of each protein. And if you know what you're looking for, you can isolate specific proteins from a cell using this technique of electrophoresis.